Hello, everyone. I'm Russ Kleinman, and I'll be giving a very short presentation today on nitrogen fixation, and I'll explain what that is, and bryophytes. Or I added a subtitle just yesterday because I love the way that Goffinet and Shaw introduced this subject in their book. They say no carnivorous bryophytes are known. What does that have to do with this whole topic? What is nitrogen fixation anyway? We have an electrical engineer here. We've got a computer scientist. We have a lawyer. We have all kinds of people in the meeting here who don't really have a solid biological background, perhaps. I don't really know. So let me go ahead and describe a little bit about what nitrogen fixation means. What is nitrogen fixation? Well, I'm sitting here breathing, as are we all. And as we breathe, we're breathing air. Air is 78% nitrogen. The problem for us is that we can't use it. We're breathing in oxygen, which we can use, and we're breathing out carbon dioxide. But we can't use 78% of the atmosphere that we're breathing in. It's nitrogen. And yet, we need nitrogen. It's part of much of what our cellular metabolism needs for amino acids, for proteins. We make amino acids, they contain nitrogen. Proteins are made of amino acids that contain nitrogen. DNA, the nitrogenous base part of DNA, contains nitrogen. All kinds of things that we as organisms need are made partly of nitrogen. Problem is, we don't have any way of using the nitrogen that we're breathing in, and we have to get our nitrogen in some other way. It has to be made usable for us by some other organism. On Earth, as far as we know, until recently, and I'll get there in a minute, only some bacteria among living organisms have been known to convert atmospheric nitrogen to usable nitrogen, usually in the form of ammonia, sometimes nitrates or nitrites. But for the most part, bacteria have been what life on Earth has relied upon to convert atmospheric nitrogen to usable ammonia, which then gets added to substances to make amino acids, which are then converted to all kinds of other molecules. Lots of organisms have associations with these bacteria that can make atmospheric nitrogen to usable nitrogen. Ferns, one in particular, I'm thinking of a water fern, I'll show you in a second, that farms bacteria and provides those bacteria with space and with perhaps some carbohydrate and gets nitrogenous, usable nitrogen in return. Peas and beans, legumes, they have nodules in their roots, as do alders, and they keep these organisms in these nodules in their roots, and they get nitrogen from them that way. Bryophytes also have associations with these bacteria and get usable nitrogen from the bacteria and provide the bacteria with carbohydrate. Do any of these organisms, including bryophytes, do nitrogen fixation themselves? No, they don't. So at no point do I want to imply that mosses are actually making usable nitrogen, ammonia, or nitrates themselves. They're getting it from bacteria, including cyanobacteria, that they have an association with. So then here are some examples of what's going on. I don't know, I think you might be able to see my cursor here. So here is Trifolium repens or white clover growing in the lawn. Probably growing in the lawn here at Western. And if you pull it up gently, because they pull off easily, you can see root nodules here. Inside of here is where the nitrogen fixing bacteria live. So the clover isn't making usable nitrogen itself out of the atmosphere. But the bacteria, in this case, a bacterium called Rhizobium, is living inside these nodules and is transforming, that's a bad word to use here, but is basically making usable nitrogen in the form of ammonia out of atmospheric nitrogen. These nodules are oxygen excluding because oxygen actually inhibits the enzyme that breaks the nitrogen triple bond, which is hard to do. It takes 16 ATPs for those of you who know how biological systems work. That's a lot of energy. It takes a lot of energy to break the nitrogen to nitrogen triple bond that is dinitrogen or N2. That occurs in these nodules. And on the right side of the screen here, this is Azala mexicana. This is actually a water fern. It doesn't look like some ferns that you might be familiar with, but it is a fern. And if you put this under a microscope and break open a few little cavities, you're going to see, again, a cyanobacterium that I've ruptured out of this cavity. All I did was put a little bit of this under a microscope and press on the cover slip with my thumb, and it ruptured the cavity that all these filaments were hiding in. And these filaments of cyanobacterium pop out. And you can see that there are two types of cells that make up these filaments. These more oval and fat cells are, again, oxygen exclusion chambers this is where nitrogenase, the enzyme, hangs out, that's breaking that triple bond and making ammonia. So Azala farms this cyanobacterium, which is actually Anabaena azalae. So this cyanobacterium is being farmed basically by the fern, and the fern gets usable nitrogen, ammonia, from the cyanobacterium. The cyanobacterium is getting carbohydrate from the fern in much the same way that the nodules, the rhizobium in these nodules, is functioning in a symbiotic way with the pea. Okay, so what about mosses then? The mosses also have, and it's not just mosses I'll be talking about. I'll be talking about liverworts and hornworts as well. To start off with mosses, mosses also have associations with bacteria, and in particular cyanobacteria. Most of these associations are with a cyanobacterium by the name of Nostoc. And in this picture, you can see a mucus cavity here. It's not really a cavity, mucus layer around the nostoc. The nostoc makes this mucus layer. The moss didn't. This is Hyophylla involuta. I just collected this a week ago in the Gila here in southwest New Mexico, and saw all this nostoc on the surface of the moss. And what is happening here, just like it's doing it when there's actually a well-defined nodule, is there you can show with radioactive isotopes that ammonia, after the nitrogen is fixed by the nostoc, ammonia is transferred to the moss, and the moss is transferring carbohydrate to the cyanobacterium. Again, in mosses, it's almost always a surface association. There's no specialized cavity or layer within the moss itself in which this is occurring. We move on to liverworts. Again, it's mostly nostoc that we see involved here. The association between the bacterium, the cyanobacterium, and the liverwort can either be on the surface or it can be within the body of liverwort. Here I have Parella. Parella is only one cell thick, so nothing is going to be inside a thallus here. You have the cyanobacterium sitting basically in a little mucus nodule on top of the surface, but still there's transfer of fixed nitrogen of ammonia from the cyanobacterium to the liverwort, and in return the liverwort's transferring carbon compounds, carbohydrate, to the cyanobacterium. This is a surface association here, but here you have a thallus liverwort, and you have nostoc colonies within the thallus of Blasia. Barbara, I think this is a David Wagner photo.
Yeah, looks like it. Very good photograph.
Yeah, useful. Here's the Nostoc colony. That would probably be internal to the thallus. And again, the Nostoc is transferring, especially in these internal nodules, probably 50 to 80% of the fixed nitrogen is being transferred to the liverwort. And liverwort, again, in return, is transferring carbohydrate to the cyanobacterium. Things that get even more interesting when it comes to hornworts. These pictures are ones I took in California. There hasn't ever been a hornwort described from the state of New Mexico, so I can't show you anything local here. Here's an anthoceras that we collected from Armstrong Redwood State Park a few years ago on Sobe Free. And within the thallus, you can see that I had to actually shoot through the cells here. Within the thallus, there's this blue-green alga or cyanobacterium right there. If you cross-section that, here's a cross-section of the hornwort, you can see there's a cavity there in which the cyanobacterium is hanging out. Here's a different hornwort, and you can see you can actually see these nodules that are just inside the thallus here, on the inside the thallus of the hornwort. Again, if you cross-section it, here's a cross-section I made, and I cut the nose off that little nodule there inside the thallus. And if you then squeeze a little bit, guess what? You can get the cyanobacterium to come out of that nodule and prove, in fact, that these nodules are holding a cyanobacterium. And again, with radioisotope studies, you can show that nitrogen is passing from these nodules into the thallus, and carbohydrate is going from the thallus into the nodule. Now, in hornworts, commonly or frequently at any rate, you don't have just a nodule. The cyanobacterium can be in a layer underneath the surface here. So you don't always see a neat nodule. And I'll leave you with one final thought before I open it up to questions. We have always thought of nitrogen fixation being solely in the province of prokaryotic life forms, bacteria. We have thought, and a few archaea, if you are really into knowing your biology. So we have thought of nitrogen fixers being only bacteria, cyanobacteria, and archaea. These are organisms that are very small, almost all unicellular, no nucleus, and no intracellular organelles. So we have never found, and never even gave thought really to, a eukaryotic organism, an organism with a nucleus and intracellular organelles that might actually have an intracellular organelle that performs nitrogen fixation. That had never been found. In fact, most people didn't think it was possible until last year, in April, when an article came out in the April of 2024 Science journal. And this wasn't a fly-by-night author. This was a multinational, multi-institutional study of an organism, and I hate to butcher the name of it, but you see it there, the marine alga Rudosphera bigelovii, a marine alga, which you can see on your screen now on the left, that contains something that they called a nitroplast, in which they have shown that nitrogen fixation takes place. Now, why is this not just an endosymbiotic situation? Why is this not just one organism inside of another? Well, it's because part of the genome of the nitroplast now resides within the genome of the alga. And when the alga divides, the nitroplast divides synchronously with it. Basically, this nitroplast probably started out as a cyanobacterium, just like chloroplasts did. They were probably in a symbiotic event that transferred some genes to the host genome, and then began to divide synchronously with the host cell until it could no longer be a free-living organism, and then it was an organelle. That happened to a chloroplast. That is also what we think happened with mitochondria. We think they were an endosymbiotic event as well. There's a lot of proof of that. Among other things, the genetic material in chloroplasts and mitochondria is circular, just like in a bacterium, not linear, like those of us who are eukaryotes. In this nitroplast, it's a bacterium that became engulfed and then became an organelle. It fulfills all the criteria of being an organelle and no longer being an organism that can be free living. So we have our first example in history, in science, of a eukaryotic organism with a nitrogen-fixing organelle, a marine alga. And so the thought I leave you with is, okay, what were the first land organisms? Well, first land organisms were bryophytes. At least that's what the current theory is. Is it possible we could find a nitroplast in a bryophyte? Can't think of any good reason why not. If you look that up and ask that question, how likely is it that a bryophyte could develop a nitroplast and become a nitrogen-fixing bryophyte? And most of the answers you'll find online, the question's been asked, most of the answers you get online are that it's highly unlikely because the associations between bryophytes and cyanobacteria work so well as it is that there's no reason to have a nitroplast. For some reason, I find that to be a little bit of a weak argument.
Yeah.
That might have been the case also with this marine alga.
Mm-hmm.
So I think there needs to be a little bit of I think someone would do well to search around and see if you can find a nitroplast in a bryophyte.
Cool.
Cool.